On behalf of the organizers, we would like to extend a warm welcome and express our gratitude for participating in our webinar Genomic Talk Series 12 with a topic about metabarcoding for biodiversity. I will make a brief introduction to our moderator, Dr. Rahadian Pratama. Moderator profile, Dr. Rahadian Pratama is a lecturer in Department of Biochemistry at PBU University, the Bachelor of Science in Biochemistry program at IPB University in Bogor, Indonesia from 2006 until 2010, and getting a Master of Science in Biochemistry program in IPB University at Bogor, Indonesia from 2011 until 2013. And he got a doctoral degree from University of Göttingen, Germany, and finished doctoral degree in 2019. Sorry. If we see the Google Scholar, he got the citation since 2017. Citation is 11, and it's index 2, and I-10 index 0, and research interest score 43.5, and citation 8, and its index is 2. With all due respect to Dr. Rahadian Pratama, Postdoctoral, the time is yours. Thank you.
Okay. Thank you, Mbak Isti. Can you hear my voice, Mbak? Okay. Hello, Rosie. Can you hear my voice, Rosie?
Yes. Hi, Rahadian.
So, how are you?
Good. How are you?
I'm very good, thank you. I'm very happy that we can meet again after our last workshop, in Bogor, Podcaston about the Bogor workshop, and then today we have quite a good opportunity to share this knowledge to more wider participants. Okay, before we start, I would like to introduce Dr. Rosie Drinkwater. Mbak Isti, can you share the screen for background? So, everyone, today in this good afternoon, I believe in Bogor it's raining, but I hope it does not hinder our activity, especially to hear the good sharing session from Dr. Rosie Drinkwater. Dr. Rosie Drinkwater is a postdoctoral researcher at Ludwig Maximilian University, Munich, LMU in Germany. She studied her Bachelor of in zoology in the University of Edinburgh, and then continuing to the Imperial College London for ecology, evolution, and conservation master degree, and then her doctor was graduated from Queen Mary University of London in molecular ecology, and now she is based in Germany. She was quite expert in biodiversity, so the topic in general topic Rosie in biodiversity if I'm not wrong in biodiversity, and I learned from her a lot in our last workshop in Bogor about how we about babi rusa in Sulawesi and in general metabarcoding, and I believe that in Indonesia, especially since I also coordinating with SGI, that the metabarcoding, whether is it the 16S or is it for eukaryote in general, it's now getting traction. So it's increasing the research, and since in Bogor, usually in the past time when we want to do some research including NGS, the option is quite limited, and we have to send the sample outside of Indonesia. And when you want to get another support, it's also language barrier and so on and so on. But now here in Indonesia, also in Bogor especially, that we have a facility that able to do the same services, and also expanding the capability of our researcher, especially in Indonesia, to progress in this area. And I would like, then I asked Rosie, can you please share with us? And then, yeah, I think it's a good opportunity. And I'm very happy that Rosie able to join us this afternoon. Without further ado, I think now let's start the season of Metabarcoding for Biodiversity. And I would like to invite Rosie.
Yep.
Can you maybe try to share your screen, Rosie, just to make sure
Yes.
it's okay? Mm-hmm.
Can you see that? And
Yeah.
is this the right one, or do I need to
No,
swap?
I think you should switch the presenter view and now it's
Okay, good.
okay. Okay, thank you. So Rosie, usually the total time is 1 hour, so you have like 40 minutes
Okay.
to present, and we have a discussion session, yeah. Okay, so
Okay, great.
now the screen and time is yours.
Great. Thank you very much. Thank you for the kind introduction and the invitation to speak today, Radian. I'm going to be talking to you today about metabarcoding and how we can use this for biodiversity surveys and in general to apply to conservation. So to do this, I'm going to talk about a couple of case studies that I've been involved in. So probably the biggest one I've been doing is about getting mammal DNA from terrestrial leeches. So this was in Borneo in Southeast Asia. And then a little side project, which was when I was trying to get DNA from dung beetles as well. I'll also quickly talk about the air DNA project that I've been working on. So this is sequencing terrestrial or vertebrate species from air. And then if there is time, I'll just quickly go on to how we're going beyond just taking species inventories using this metabarcoding data, both from a sort of modeling analytical angle and also a technical angle as well. So you'll probably be familiar with this, the difference between metabarcoding and barcoding, but just so everyone's on the same page, DNA barcoding is sort of targeting a single species with a specific primers that are for a common marker for that group. So for bacteria, 16S, or for mammals, CO1. Metabarcoding is where we take a mixed sample and we're targeting the whole community of species that are in that sample using primers that match to many different species. And normally the input for these experiments is environmental DNA or eDNA. So the broadest definition of environmental DNA would be any nuclear or mitochondrial DNA that's released from an organism into the environment. And this was first used a very long time ago in the microbial communities to describe bacteria and things in soil and water. But now we've sort of adapted it to work for vertebrates as well. And we can get this DNA, the eDNA, from many sources. So it could come from the organism, the target itself, from dead targets, from skin cells, from gametes, basically anything, any DNA in the environment. So the ecological community has a lot of different techniques for surveying biodiversity, camera traps, mist nets, things like this. So what are the benefits of using DNA as well for surveying these communities? So it's faster in the field in most cases. So collecting water samples tends to be quicker than going out and setting up camera traps, leaving them out for months and going back multiple locations. It allows you to remotely identify the target species. So you don't need to do any identification in the field. It's all done bioinformatically with the computer. And because of this, you require less taxonomic expertise because it's done through reference databases and reference sequences. It gives us access to rare species. So species that you would maybe otherwise not see necessarily, or that would need a really long and comprehensive field campaign. So if you're thinking of rainforest birds or mammals, you would need to repeatedly sample a site with cameras or mist nets for months on end to make sure that for the rare species that you were getting enough detections to see it, to make a decision whether it was there or not. And then because of the speed of collecting all the samples and all these benefits, it allows you to then maximize the geographic area that you can cover in the field. So why is all this important? Well, in our current biodiversity crisis, we need to be documenting species and assessing ecosystems as quickly as we can. So there's many estimates, but this figure just shows the IUCN risk categories. And in orange and red, they're the categories most at risk, the threatened categories. And you can see for mammals, you have nearly 30% of species are already in these risk categories. And that by many estimates, we could be actually experiencing between 200 and 2,000 extinctions per year. And so while traditional surveys for ecologists are absolutely vital, we still need to be doing traditional ecology. Biodiversity surveys using DNA give us this extra tool for rapidly surveying these ecosystems that we think are at risk. And as time moves on in the field and increasingly in the lab, we're actually getting faster and faster at processing these samples. And it's also getting cheaper. So we can get eDNA from many sources. Soil and water probably are the most commonly used sources of eDNA. And water samples as well, in some cases have been used so for government national surveys. So in the UK, for example, we have the great crested newt, which is in endangered or threatened species. And the government environmental agency will take eDNA water surveys to prove whether the newt is in the pond or not. You can also take bulk insect traps. So this is where you just collect everything, all the insects, and then you sequence what you've got. And scats or guano is also quite a common use. But there's also many novel sources of DNA or niche sources, let's say. So some groups in Scandinavia have been working on detecting predators from footprints and urine in the snow. You can get prey species off spiders' webs. So using them as nets, essentially, for biodiversity surveys. You can wash invertebrate DNA off leaves as well. That's also recently happened. And the two I'll be talking to you about today is invertebrate guts and air. So essentially, maybe we're only limited by our imagination from where to get DNA from. So there are some considerations we need to take into account if we're going to run these metabarcoding surveys with the intent for surveying biodiversity. So we need to understand the biases of the method, as with any sort of ecological survey method that we use. And there's this concept called the ecology of eDNA, which was summed up in this paper, which is a bit old now, but I think it's a really useful paper, and to get you thinking about the things that affect the DNA from source to sequencing. So what happens on its journey? So you have to think about the origin of the DNA. Where did it come from? Is it actually from your target species, or is it some kind of secondary DNA? So for the prey that the predator's been eating, or some sort of scavenged DNA. Its state, do you find it free in the environment, or is it still within a cell membrane? Transport is a really important one. So what you're detecting, is it actually from the source, or has it been moving? Is this a biological movement? So maybe think DNA down a river, or have humans been moving it in some way, which is also a common issue. How has the environment been interacting with it to change the degradation state of the DNA? So has it been sitting out in where the sun has been shining on it for ages? And this is all before you even get into the lab. So once you get into the lab, then you need to consider things like contamination and fragmentation and inhibition. We also need to consider things like what genetic markers we're going to use. And you might be following previous work where they've kind of shown you that these primers, these markers work for identifying these species, or maybe you have to get a load of primers and test, start from scratch and test it yourself. And this will depend on whether you want to target the whole community or a specific group or species. Are you going to take individual samples or are you going to pool samples together? So if you take repeated surveys at the same site on the same day, which is kind of the model of one of my projects, it didn't really make sense to do them individually because it's the same information. So we pooled everything together to be more cost effective. And reference databases. So you're only as good as your reference. And this is mainly because with metabarcoding, we assign the sequences to operational taxonomic units, which are OTUs, or amplicon sequence variants, ASVs. So OTUs are clusters of sequences based on similarity. Sometimes an arbitrary cutoff of about 97%, whereas ASVs are a bit more stricter and they just show single variants. But we basically take these unknown OTUs or ASVs, you compare them to the reference. So if you're using particular groups, there might be curated databases for you. So the BOLD database, the Barcode of Life database, has a lot of CO1 markers. There's 16S, the microbial databases are quite good out there, and the fungal database for ITS as well. Okay, so I'll just go on to the first case study, which is regarding leech blood iDNA. So what is iDNA? I've used this term, which I use to mean invertebrate-derived DNA, but some people like ingested DNA, but whatever people prefer. And this picture just shows kind of the steps that you take. So you're wanting, your target is these mammals in the rainforest, but the leeches are at a higher abundance, so you collect a lot more of those. You take them back to the lab, you extract the DNA, you sequence the DNA after amplification for the target. You sort the sequences bioinformatically, and then using your reference database, you identify these mammals all without seeing them. So this is why it's a remote survey technique, because you actually never need to lay eyes on these animals to know that they were in the site you were surveying. So as well as all the general benefits that I mentioned before, specifically related to iDNA, it's a lot easier to catch your invertebrates than it is to catch or see your vertebrate. So the leeches, the flies, the mosquitoes, they live at higher abundance than the target mammals or animals. Many of them are assumed to be generalists, and I say assumed because it's not often known a lot of the time, but if they are generalists, this makes them good proxy samplers of your target, really. So if the mammals are in the site, they're going to be just surveying and eating the mammals for you, and therefore giving you a good sample. It's a non-invasive technique. This is from the point of view of the mammal, because from the invertebrate's point of view, it's quite invasive. And again, this gives you access to rare species. So if your sampler, your invertebrate is a generalist, it's just sampling everything. It's going to pick up those rare species as well. But a really important question is, which invertebrate do we use? So this is the carrion flies or the blowflies, and they've been used quite a lot in West Africa for identifying mammals. People have also used mosquitoes. And the additional benefit of using mosquitoes is you can ask questions, epidemiological questions as well, as they're a disease vector, which is the same with the sandflies. So the sandflies have been used in South America. So these are the popular fly samplers. So there's also the tsetse fly as well, these have all been used quite successfully to identify vertebrates. We can also get DNA from dung beetles and terrestrial leeches. So I'll talk about these in a minute. And really, as with most things, which one you decide will depend on your research question. So sometimes the decision will be made for you. So you have to choose an invertebrate that's at high enough abundance in your field site or your study area. You need to know a little bit about generalism of your invertebrate to make sure the sample is appropriate for your question. So if you want to target a specific species, you need to make sure your invertebrate actually feeds on that species. And if you want the community, you need to make sure it is at least thought to be generalist. This will all depend on the invertebrate's feeding ecology. The way in which you trap for the invertebrate is also really important, because this adds in different biases into your assumptions about where and when you found the mammal DNA. So for dung beetles, you can do pitfall traps. Carrion flies, I think a lot of the studies were done with nets over different carrion. But for things like leeches and in some cases mosquitoes, you kind of just have to use yourself as bait and wait for them to come to you. And finally, something that's maybe a little bit harder to know without some sort of pilot studies is something that I've called a feeding window, but this is basically how the DNA degrades over time in the gut of the invertebrate. So this is trying to get some kind of timestamp on when and where, on when your mammal, let's say, was found in that area. So all these considerations, if you think about the ecology of eDNA before, this is kind of related, but more specifically for iDNA. But we need to think about how the DNA changes from being from the invertebrate feeding on the mammal to us sequencing it. So these are the samplers of my samplers of choice, the brown leech and the tiger leech. So they're both terrestrial leeches that you find across most of Southeast Asia. They're from the family Hemodipsidae. Yeah, they've got a Southeast Asian distribution. The terrestrial leeches are actually only really found in Southeast Asia, and a couple of strange species that are found in Madagascar and Australia. They tend to be, when they're present in the humid tropical forests, they tend to be at high abundance. And they're quite easy to catch because they come to you. You don't even need to really trap for them. And they're assumed to be generalists. Well, at least the tiger leech is assumed to be a generalist species. So why did we think we could do this? Well, the first paper that got DNA from leeches was in 2012 from this group in Copenhagen from Ida Schnell. And they were looking for this mammal, which is a really rare antelope called the saola that lives in Vietnam. And unfortunately, they didn't actually find the saola, but they found some pretty rare and unrecorded species, so including the serau and the Annamite striped rabbit. So also with this paper, what was very important is they conducted this feeding experiment. So this is the feeding window that I was talking, that I mentioned before, where they fed, this is actually a medicinal leech, European medicinal leech, but they fed this leech in the lab goat blood and took different samples at different times and measured the amount of DNA copies in the blood using qPCR. And what they found was this decay in DNA copy with time. So this shows us that the DNA is degrading in the gut. But if you can see, you can still get detectable amount of DNA even like 80 days post-feeding. So this increases the chance that when you took a leech out of the forest, it would have the DNA of its last meal in its gut. But also it means that the time window of when the animal was around is actually could be quite long. So the aim of our project was to see if we could characterize biodiversity across a habitat gradient using leech DNA. So can we apply leeches to an ecological question about whether diversity is different in different land use habitat classes? This was a really big project. It was part of my PhD. So this was done at Queen Mary University in collaboration with UMS in Sabah. And there was lots of people involved, including technicians and all the research assistants who helped me collect a lot of leeches in the forest. So I'm pretty sure you all know where Sabah is, but this is Sabah in Malaysian Borneo. And all the field work was done here at two different field sites. So first, the Danang Valley Conservation Area, which is here. And this is a primary old growth rainforest. There's very high levels of biodiversity and low levels of human impact. And then the complete opposite was the SAFE project. So this stands for the Stability of Altered Forest Ecosystems Project. And it's a large-scale forest fragmentation experiment. So they took a patch of forest and they were slowly leaving patches of forest in the land as they converted the land to oil palm plantations. And so what resulted is a mosaic landscape which contains multiple different habitat types, going from high quality selectively logged forest through all the stages of logging until the final land conversion stage of oil palm plantation. So this is Dannon Valley. We've just got very high trees. We've got a closed canopy. And actually the tallest tropical tree that's been measured to date is inside Dannon Valley. And you compare it with this. So when I was at the SAFE project, active logging was actually happening at the time. So as you go through the landscape, there's roads, there was trucks, there was these landing sites, and a lot of infrastructure for logging. But also there was this. So at first glances, it might not look so good, but if you see, the canopy is almost closed. So this is higher quality, selectively logged forest. So there are no tall trees here. They've taken out all the high value timber trees, but actually in comparison, the damage is relatively low, and the microclimate, which is very important, is actually relatively stable and comparable to the old growth forest. Because we're comparing it to this. So these sites are the heavily logged forest sites, and these have been salvage logged. So basically all the trees have been taken out. There's still these scars in the landscape. If you leave the land for a little bit of time, this is what happens. So it's the vegetation starts to grow back, but it's all grassy pioneer species. There's no trees yet. Eventually they'll come, but not yet. And then it goes all the way to oil palm plantations. So this was the major land use in the area. And we know from camera trapping in the region that some groups of mammals really do actually use the oil palm plantations, even if they're just moving through, but it's quite well traversed. The problem is you don't get leeches. So the microclimate, if you've ever been into an oil palm plantation, the microclimate is very hot and dry, and it's too inhospitable for the leeches. So unfortunately, we can't make this most extreme comparison between the highest quality old growth forest and the oil palm plantation, which is again a consideration for your study design. If that was the question that you wanted to answer, leeches would probably not be your sampler that you would be able to choose. Okay, so I've gone across all these different habitats. I've collected leeches at different fixed locations, and I take them back to the lab. We run a very, at the time, quite a standard metabarcoding pipeline. And I'll just point out some key points that I think are interesting. So we actually extracted the leeches and then pulled them together for the PCR. And in this way, we did this based on site and survey, but we did this to obviously maximize the amount of be cost effective with our resources, but also maximize the amount of detections that we might be able to find. We used a special set of primers which actually target 16S, but are based on ancient horses. So they're very small fragment primers, and they target a very small fragment of DNA. These were sequenced with Illumina. We get the sequences back, we do standard quality filtering of the reads, and then we did this study based on clustering of OTUs. So these are the operational taxonomic units. They stand for a kind of proxy of species or genus, because the sequences are clustered based on similarity. There are a few issues with this method, with just clustering of OTUs, because you can artificially inflate your biodiversity estimates by counting OTUs as real, but actually they're just arising from sequencing error. And so we use this program, Lulu, which it runs a post-clustering filter on the OTUs and basically gets rid of a lot of the OTUs that you find just by sequencing error alone. We then identified the mammals, comparing them to our reference database. And in some ways, we were perhaps a bit blessed in comparison to some metabarcoding projects. So we actually, people know a lot about mammals, for one, and in this place in Borneo, the mammals have been really well studied. So we knew kind of exactly what mammals should be there. And we also had a lot of the reference sequences already. The ones that we didn't have, we actually sequenced. So we got some extra civets. So there's a high civet diversity in that region. So we got some of the extra civets from the museums. We added in representatives from related taxa that weren't in the that we didn't have reference sequences for, and we added in common contaminants, which is also quite important. And then all these sequences were taxonomically assigned using OBI tools. So can we do this? Can we characterize the biodiversity across a habitat gradient using the leeches? So this is what we found. So this is a frequency of abundance plot. So across the x-axis is the detection, so the number of times a species was detected. And along the y-axis, this is actually to genus. Because the fragment was quite short, so a few of the taxa, we couldn't discriminate between two conspecific species. But we find the highest detection rate for the most common mammals. So this is a good proof of concept. So we get a lot of bearded pig, we get the muntjac, the samba deer. We also get the hystrix. So this is a porcupine, a large porcupine. And this is one of the taxa that we couldn't discriminate. So there's actually two hystrix porcupines that live in the region. And both are actually quite abundant, at least based on the camera trapping data. So we just, to be conservative, we've just left everything at genus level. We also detected a collection of rare species. So we detected pangolin. Pangolin is probably one of the world's most trafficked group of mammals. We found the elephant, the Bornean pygmy elephant, and the sun bear, which are also species of conservation concern. And also reflecting the known diversity of civets, we found 4 genera out of a possible 7. So a few of the civets are super rare and they basically have never been seen. So out of the 5 most common, we were only really missing the binturong. And the binturong is perhaps not surprising considering how arboreal a species that it is. We then calculated the diversity at different sites. So these plots, the colors, so dark green is the old growth forest, light green is the twice logged, which is that selectively logged kind of high quality forest. Purple is the heavily logged forest. So these are actually based on something called hill numbers, which I won't go into, but you can think of it as the equivalent of species richness. And if we look at the rarefied diversity, so this is when you take the diversity calculation but account for sample size, you can actually see that there's not much difference between the different habitat types. And if we look at another metric, which is a hill number equivalent to the Shannon Diversity Index, and again, we look at the rarefied diversity, there's very little difference between these habitat types. And this is a good thing because heavily logged forest, the worst, the trashed forest, is at real risk of being fully converted to oil palm. And actually by proving or showing that there's sort of a consistent similarity between the heavily logged and the higher quality logged forest, we're assigning conservation value to these heavily logged patches, and perhaps they would stay as forest, if not really low quality. We also took a look at beta diversity, so community level differences. So these are NMDS plots. And if you look, each point is a site. And the distance between the points is the difference in community, the relative difference in community between the sites. And what you can see is actually that the heavily logged forest sits within the higher quality logged forest sites, which is good. It shows that it's like a nested subsample of the communities that you find in the logged forest. And actually, it's not a complete turnover of species between these sites. So next, this is just a quick one, but using the same sort of pipelines, we wanted to see if we could swap in dung beetles for the leeches, to see if we could use our high throughput method for the leeches to harness some of the benefits that we get from dung beetles over leeches in their ecology. So what are these benefits? So number one is probably that you can trap for them. So even though with the leeches, it was a systematic survey, you're still waiting for the leech to come to you. Whereas with the dung beetles, you can put out traps, collect them after 24 hours, and they can be in fixed distances apart. And this means there's potentially less or different biases involved in this sampling method. They're less habitat restricted, so you can trap for them in the oil palm, which is good, at least some of the species. And they have a really wide geographic range. So you can find them in all continents except Antarctica, which then opens up the possibility of being able to compare different sites across the globe. Whereas with the leeches, you are kind of restricted to Southeast Asia. They have a range of feeding strategies across all the groups. So you can get really specific ones that just feed on one taxa, or you can get complete generalists. And eventually the idea is that we actually use these as indicators of mammals. So if we can find some sort of structure between the presence of a beetle and the presence of mammal DNA, maybe we can even take out the metabarcoding step at some point eventually. But the problem is the DNA here is like twice digested, so we're thinking it's even more highly degraded. So we wanted to run some preliminary pilot experiments first. And we did this using kind of trying to emulate the leech feeding window experiment. So we collected these beetles here from the field. So these are just, there's nothing special about them, just that I'm not an entomologist, and these are the biggest and the easiest to collect and identify. So we collected these ones, fed them on a set amount of cow dung in the field lab. We just dissected the guts at certain time points, and we did a similar qPCR experiment to look at this window of DNA detection. And as a nice sanity check, we see the same sort of decay of DNA over time. But if you can look at the feeding time here, the leeches was in days, and whereas the beetles is in hours. So actually, this is perhaps another benefit as well for the beetles, is that the turnover of the DNA in the gut is actually faster, a lot faster. And so when you get a detection inside a beetle gut, you can say that the mammal has been in the area within the last 3 days, 4 days, whereas for the leech, your window is much bigger than this. However, you might be more likely to collect, let's call them empty beetles with no DNA in their guts. So this is a kind of trade-off. So obviously this was just a very quick pilot study, but we did find the same common mammals at high abundance, so bearded pigs, samba We also found evidence perhaps of the banded civet, but obviously if you look at the read count, this would need some validation, which is actually what we're doing with a team in Singapore. So hopefully we're going to expand this work, because beetles seem actually like a really promising sampler for iDNA. Okay, so I'll just talk about the last case study, which is air DNA. So our question here was, can we sequence terrestrial biodiversity from the air? So this was the work of one of my PhD supervisors, Beth Clare, in Canada. And if you look at the early eDNA papers, a lot of them say, at least for the terrestrial or the vertebrates, a lot of them say you can get eDNA from any source, so from water, from soil, and often they say air. But if you do some digging, this has never actually systematically been done before, at least for vertebrates. So we started with a proof of concept. And our idea was to try and identify this guy, which is the naked mole rat. And we chose the naked mole rat for 2 reasons, because we were at Queen Mary's University in London, and they have a colony that they house there. And also we needed something exotic. So we needed something that we could definitely be sure that the DNA is coming from this creature. So it couldn't just be like a dog, or obviously not human. And so we went into, or a colleague of mine went into this room and filtered air over a pump. We amplified this using 2 markers. And because of the way this experiment was, we tried Sanger sequencing, and we detected just mole rats. The results were we detected just mole rat and human DNA. So this is a really good proof of concept that the DNA in the air, in the room, was just humans and mole rats, which are the only two animals that use the room. So next, the idea is we needed to build on the system, but we needed to maintain the controlled kind of settings because we need to be able to trace back where the DNA is actually coming from. So this was happening during COVID, and Beth had this great idea to go and test the air in a zoo. So the zoos were closed, so there was no people, so hopefully reduce. And this is Beth here just using the air pump to suck DNA from the air. And this is the pump which just sits, and it's like a peristaltic pump. It just takes the air in outside the dingo enclosure. So the aim was to see if we could identify the exotic zoo species against the background of local species. And this is what we found. So the larger circles are the higher read abundance. And this is a map of the zoo. And we actually find that the target species in green, at each enclosure, we find a higher read abundance of each target species. So at the gibbon enclosure, we find the gibbon, the binturong DNA, meerkat DNA, and so on. But we also find these species DNA across the zoo. So there is some sort of DNA decay with distance. And we saw some kind of relationship with this. It was not significant, but there was some kind of pattern. We also found local species. So this is the European hedgehog and the grey squirrel. And these two are quite interesting because the European hedgehog is a threatened species in the UK, and the grey squirrel is an invasive species in the UK. So this shows you some kind of applications that you might be able to use these techniques for. We also found dietary items. So outside some of the carnivore enclosures, you had a really high read abundance of things like chicken. And after checking with the zookeepers, this is because that carnivore was eating the chicken. So the last piece of the puzzle was to go and test this truly in the wild. And this was done by Nina Garrett, who's Beth's PhD student in Canada. And they went to Belize. They went to this natural system in Belize to look for bats. And this is because Beth is actually a bat biologist. So she knows the bat community in this site in Belize very, very well. And bats live in caves or tree holes. And so we thought the first time we're doing this in the wild, it would be good to maximize our the enclosed. And like I said, Beth knows these systems, so there's a really good species inventory. And we actually found that we could recover all the bat species we thought were there, which is good. And we also documented one of the missing species. So the ecologists at the site thought that there should be a bat species that they've never picked up in the nets. And actually, it's obviously roosting in the caves and the tree holes, but it's avoiding the nets, which is quite common. But the air DNA actually detected it. So this is a really cool application of this technique. So if I have time, I will just quickly go beyond inventories. So all the examples I've given you is we've been recording species, so cataloging species names and locations, but the field is moving a bit. We need to move beyond this. We know we can do this now. So I'll talk to you first about something that I've been applying occupancy models to iDNA. So at the moment, you just get a detection, non-detected or detected. If we use occupancy models, which is a modeling technique used for camera trapping, you can, from your repeated sites and surveys, you can build up a detection history. And from this detection history, you can get a bit more than just detected or not. So if the pig, let's take this example, if the pig is in the area, the camera or the leech is going to detect that pig. If it's not in the area, the camera or the leech is not going to detect that pig. But a lot of the time you actually have this situation where the pig is in the area, but neither the camera or the leech actually picks it up for whatever reason. And this is where occupancy models come in, because you distill this detection actually into two probabilities. So either occupancy probability, which is the probability a species or DNA or its DNA is at a site, or detection probability. So this is the probability a species or its DNA is detected or sequenced, given it was present at the site. So they're related to each other. And these models can get really, really complex. And there's a lot of factors that you can build in here, but I'll just show you one that I think might be relevant to us. And this is when you include covariates into the model. And in my case, I was including technical covariates, so things about PCR, DNA concentration. And if we just look at the probability of detection here for bearded pig, samba deer, and muntjac, you can see that the probability of detection for bearded pig and samba deer actually increases as we increase DNA concentration. But for muntjac, this doesn't have any effect. And so the benefit of using occupancy models in this way is we get more nuance from our detections, not just detected, non-detected, pull out some really species specific, because they could be technical or environmental, like forest cover, for example. And finally, I'll very quickly just say that people have actually been trying to get population genetic level data from leeches, which I think is really cool. So this group in Berlin, they went back to the Annamite striped rabbit that they found in Vietnam. And they did this targeted amplification of multiple mitochondrial fragments to reconstruct the genome. And this is really important from a conservation aspect because the rabbit has no museum specimens, no genetic data, no information really. So how do you build a conservation program for it? And what they found is just by looking at genetic diversity at different sites, so these colors are different sites, is there's actually very low genetic diversity between, difference between the sites. So therefore, this might help inform your ex situ conservation actions. And finally, the group in Copenhagen have also been doing mitochondrial baits on leeches. So if you have enough time and money, this is something you could also do from leeches, where you basically are trying to sequence the whole mitochondrial genome. And they found it was very successful. And basically, this is where the pools of flies and leeches fell out on their tree for the non-human primates. And they found that this aligned pretty well with what they thought. Okay, so hopefully I haven't been too long, but I hope I've been able to show you that these novel sources of DNA are really useful for rapid biodiversity assessments, and that this is becoming more and more crucial in the biodiversity crisis and with our human modified world. These methods seem to be quite good at detecting common species as well as these rare species. And we can use these diversity statistics to look at how good habitats are. But we now know that we can do this. We can take an invertebrate and get the DNA from inside it. So how do we work to go beyond this? So this could be combining with occupancy models, other different methods, or different techniques. So things like mitochondrial baits, for example. So yeah, that is everything from me. Thank you very much for listening, and I'm happy to take any questions.
See, it's very fascinating, and there is already a lot of questions.
Oh.
Yeah, I just want to ask, do you able to see the question and answer feature in your Zoom? Or should
Yes.
Okay, yeah. Then I think because if I have to read again, then it takes a lot of time. So maybe you can
Yes.
directly answer. The first one from Raisa Tatum from Indonesian University. She's
who I also met in Bogor.
yeah. This about this sequencing eDNA, the average reach length, and the other primer that you use. And this question also in line with the question from Fima Inabui, also asking about the marker. Is it only CO1 from your leech DNA, or you have another marker applied? If you have like a journal or the publication for the pipeline, then maybe it's also good to share this publication.
Yes, yeah. So when it comes to Read length, this is going to be to do with which kind of sequencing you did. So it probably would be, mine was 150 base pairs, but this was the type of sequencing. And I personally would say that multiple primers and markers are always better. We did this a long time ago. So I was doing this in 2014-15. And at the time we had only one marker that was working. Our samples, we couldn't get other markers to work. But now, if you look at the newer papers, the more up-to-date papers that have been coming out, they have been managing to get things like CO1 and Site B to work, and for longer fragments. So this possibly says something about the way we were storing the samples, that they were degrading more over time. But the benefit of doing multiple markers is really that you can make confirmation between the two. So if you have a read that you don't know doesn't assign very well to a certain species, if it does in one marker, that would give you more confidence that it was that species compared to the other. This worked quite well for Borneo because our reference database was very good. We had a lot of species where they were the only one in the genus in the area. Or if they weren't, it was like a very rare endemic species versus a very common species. But yeah, I would say multiple primers and markers is definitely better for your confidence in identifying the species. It helps.
Okay, so that I think also answer the question from Eki. So Eki was asking about he tried to amplify the DNA. So it's specific primer from the 1 liter of seawater, but apparently the eDNA concentration is quite low. Yeah, this is from sea, yeah, so maybe it's a bit different, but yeah, I think we have several study about this also. And what should we do if the number and the copy of the DNA is quite low? So it's maybe not.
Yeah.
Yeah.
So I think, I mean, the problem you have with sea water is that the sea is really big. So you're going to, the chances that you got a liter of seawater and there was fish DNA in it is probably quite low. The DNA that you're seeing, so if it says the concentration was 30 to 70. I mean, there could be, well, there will be millions of other things in the seawater, right? So, I mean, my first thing to do would be to test some other primers that have a more, a wider range from like bacteria or other sort of plankton, like this kind of thing, because you'll definitely get those. And I guess with fish, I know people in Copenhagen or in Denmark are definitely doing fish from seawater. So people can definitely do it, but they might have to take a large amount of seawater to get it. Yeah. And sadly, also, a lot of these things, if you measure the DNA and look at the gel band, and this is quite hard to get people to trust you, but sometimes if you just sequence it, you will get results because the sequencing is actually really sensitive. But the problem is you have no evidence to then show someone and say, I think we should sequence this sample. But often it is the case, you get no band, you have essentially the qubit says no DNA, but you sequence it and there tends to be DNA in there.
Yeah.
But that's hard to convince your supervisors and things to sequence them.
Yeah, I think the minimum number of samples, it's also important, to obtain sufficient, I will not say large DNA, but sufficient DNA for sequencing or for amplification. Otherwise, you cannot do anything. And I think that's also related with the question from Unique Lasiana. It is in Indonesian, but I will translate for you that she was asking for any minimum standard for the number of samples that will be used. In my case, I will always say for those who work with 16S, like metagenome sequencing with 16S primer, then you have to, let's say, like 3 biological replicate, because not only for the sequencing process, but also for the statistical analysis. When you want to, let's say, do beta diversity, you only have one or two per category, per groups, then you cannot do calculation.
Yeah. Yeah, and a point that's really important, I think, is that often the sampling is the thing that you can't repeat.
Yeah.
So in the lab, you can do as many PCRs as you can. You can redo sequences. You can redo a lot of the lab side, and the bioinformatics you can keep redoing. But once you've taken that sample from the environment, whatever it is, that's it. You've done it. So I would always say more is better as well. And I would say 3 is the minimum as well. I mean, that's a kind of rule of thumb that I'm not sure where it came from, but it tends to be, at least for the occupancy modeling, if you have a detection, a non-detection, and a detection, that helps you statistically understand whether it's a true detection or not, or if it's no detection, detection, things like this. So yeah, 3 is actually, I would say, the minimum too. But I know that when they are doing, at least for the UK national surveys of the newts, they're doing like 12 PCR replicates, and they do like quite a lot. So I think, to be sure, like really sure, more is always better, which is
Okay.
yeah.
Okay, so I think the next question I will just group it. It's more to the technical, like the software that you use, and also so first one is the question from Maulana Malik Nasrullah, that he was asking the standard deviation in the hill number slide is strangely uneven. Is there some explanation in your presentation that it's kind of like that?
Hill numbers. So I'd have to think very hard. I think it's probably to do with the sample sizes are quite uneven in this group. So we have like a high, very high numbers of samples from the heavily logged forest, but not so many from the old growth forest. But to be honest, I would have to go back to my own paper and have a look.
Yeah, that's for sure. But how about the measure for diversity, Rosie? Because we know we have like Shannon, Simpson, and so on. So this is related to the Konita Hanifa question. She was asking about the Gini-Simpson index. Is it also necessary if we use like 2 diversity measure, not just one, or is it like it can strongly correlated, so we have like more solid conclusion for this?
So I think you should often use a collection of diversity indices because they are all testing slightly different things. So if your species richness is just counting species, Shannon is accounting for things like abundance, and Simpson, I think, is accounting for rare species. And so the good thing, if you start looking at hill numbers, is they are very related to this, the Shannon-Simpson index. The traditional indexes, they're basically the same, but they're just calculated in a slightly different way, which allows you to make these comparisons between them. Because strictly, I think you're not meant to compare these traditional ones. But you can get this profile, so you can look and see like, oh, well, the differences were only when it was counting species richness. And if you get more similar Shannon diversity indexes, it means that the differences was probably only the rare species, that the abundant species are quite similar. And then for your sites, that maybe means something, you know? So if it's only the rare species that are different, that's kind of what we would expect anyway. So then maybe our conclusions would be one way or the other.
Yes, that's true. Yeah, I also often have to look again because there's a lot of indices for the everything. But most used to there's a present absence, and then the evenness, richness, we have to look more.
I mean, and that's a good point. It actually depends on your input data. So if you do only have presence absence, you can often only use a certain type of index. Whereas if you have counts and abundances, it's a different set of indexes.
Yeah. Okay, and then I think the next question would be more into the tools. Yeah, I look into the from your presentation that you use Lulu for the OTU correcting the OTU. Yeah, OB tools, OBI tools, and some also R package like maybe you can use Vegan. Yeah, but are there any tools that you can recommend? Yeah, maybe yeah, because I think there's a lot of people asking for the bioinformatic pipelines. So maybe you can
Yeah.
inform them in general what you use for this.
Yeah, so for the Leach paper, we were using OTU clustering using, if I'm hopefully not mistaken, something called SumaClust,
Summer class.
which was the actual algorithm that clusters the sequences together. And then BLAST to compare the OTUs. And then Lulu is really cool because if you look at OTUs and you look at the differences between them, you can see the sequencing error. Sequencing error is just like one base change, and then that one base changes, and then this one base changes. And so it kind of like spreads out from an original OTU. And this is what Lulu does. It finds the parent OTU and collapses all the other OTUs into this one. So that's a really good for the air DNA, we used DADA2 and we did it with ASVs. Again, using BLAST to compare to our reference, I guess, as well, because DADA is very set up for sort of microbial stuff, and so often we can use these pipelines and then you have to take it out and then use BLAST to compare to your own reference database that you made yourself.
Okay. Now, so for those who interested for this, the tools that Rosie mentioned, I look one by one and I copy and paste the link into the chat Zoom so you can look more into the tools. Yeah, because for sure the time is limited, so we cannot describe the whole pipe.
Yeah.
But I think from this you can have a look, yeah, and try to explore the tools that needed, yeah, and it's really yeah, I can imagine Rosie, because the first time I also learned bioinformatics, for me that have no background for this, it's quite not scary, but quite confusing since tools that we can use, the pipeline and so on, yeah, and for them.
I know we're running out of time, but the one thing I would say is that there is a lot of tools out there, and that actually, if you find one that works for your data set and your data, then use that. It's quite overwhelming when you look at all the compared this one, and as long as you understand what the tool you have used has done, I think that's totally valid to use that.
Yeah, great. Okay, question. We have to limit. Yeah, I think I will just choose another.
I could answer any questions after if that's helpful.
Oh, maybe we can also open chance for those who want to ask directly to you. Yeah, I think we can let them raise hand. If you want to ask directly, please raise your hand, and then we can set you to able to talk, yeah, via your microphone. If there's anyone, please raise your hand, and then I can set you to be included, yeah. So while waiting for anyone want to raise their hand, so I will just answer a question from anonymous attendee. So this method that Rosie already mentioned can detect animals or mammals or animals, yeah, mostly, yeah, that we think that already extinct, but it's still there, yeah, because maybe we don't, yeah. I think in Indonesian news there is several animals that we thought already extinct, but actually still there, albeit in lower number, yeah, for sure. Yeah, yeah, that's Also, like Sumatra rhino, which is
Yeah.
10 in the conservation site in Waikambas, only 10 individual. But if we look into like the fecal sample that already scattered in the land, then we do some sequencing with the right marker, maybe we can also detect this rhino. There's also some possibility, yeah, we can do. Yeah, I think
Yeah.
so the last one is more into the like experience in your research, Rosie. So in metabarcoding research, what is the most challenging process for you? This is question from Rehan Nata Dilandes. And is there any critical aspect that we should know based on your experience? It will not be like, like you cannot learn this from textbook, but when you learn firsthand that, okay, so what we should pay attention most or something like that. Yeah.
Yeah, that's a tricky question, but a good question. I think most of the time, and you probably all feel this, is that you do an experiment and you get the results and you go, oh, why did I collect the samples in this way? And sometimes you wish you could go back. And I would say, like I said before, I think sample collection is really important. That it's quite hard to imagine whether you should go to this site time and this kind of study design layout. So that would be challenging, is trying to figure that out. And also, I think understanding things like contamination and inhibition, like PCR can be really tricky. And can work one day and you're really happy, and then the next day you're like, why is this not worked? And so like having an understanding of like technically what's happening, I think, and that is just trial and error. I think you have to make some mistakes to figure out how to do it. Or figure out what went wrong.
Yeah, the PCR process. One day it's working quite good. The next day,
Yeah. But that's metabarcoding, right? If we all worked with mice from a lab and we had perfect tissue, PCR probably would work every time. But we do have such variable samples that it just every time can be really, really different.
yeah. Okay. One that raised the hand. Fima Inabu is.
Hello, can you hear my voice?
Yes, we can hear you.
In the past, hi Rosie, how are you? Can you hear me?
Oh, yes.
Yeah, thank you for your presentation. Many good knowledge and inspiration, especially the one from getting DNA from air. I almost can't believe it, but apparently you get good data, so it works. I had some experience using difficult samples, but air is different story. So, would you mind just sharing more about it? Like, so I'm sure you did not go through DNA extraction, right? So you go straight from taking the air and then go directly to PCR reaction.
No, we did the DNA extraction. So if you take I think when we did it the first time, we were kind of just using one of those, almost like those handheld vacuum cleaners. But eventually we sort of graduated to using the Derivex pumps with the filter inside and pumping the air over it. And then you take the filter and you submerge it in lysis buffer.
Okay. And then did you get good concentration? Did you measure I'm pretty sure it's
yeah, the concentration wasn't great because obviously you've got to it wasn't wonderful, but we were able to get enough DNA to sequence to get enough sequences. But we did a lot of pooling in this, at least in the Belize bat experiment. There was a lot of samples taken at the same site and a lot of pools put together, and that increases your detection rate massively. So if you just took one filter, chances are you wouldn't get what you want.
You did some like concentrate the air, kind of like
Yeah, not directly. We concentrated it by multiplying the samples, so multiplying the filters.
Ah, I see. And what is the range? What is the range of your concentration that can be sequenced with?
I would say we might not have even measured the concentration, as in we would have just sent them for sequencing regardless, I think. Or we measure a few concentrations to check the extraction's been working, and then because it's very unreliable, just measuring the concentration of your DNA extract is it's not a very reliable measure.
Yeah, and then you went through the NGS, you get good result.
Yeah. Yeah. Oh, good for a metabarcoding. There's a lot of non bats in there.
Wow. Do you have the paper that, I mean, for that air?
Yeah, can you still see the screen? Is that still on?
Just wondering. Interesting.
Yeah, here, it's this one. Garrett et al., POJ.
Which one?
It's just at the bottom. I can write it in the thingy, in the chat.
Oh, yeah, I see it now. Sorry, it's in the bottom, yeah. Okay. Okay, thank you. I think that's all from me.
Thank you.
Best luck for your future work.
Thank you, Fima. I think yeah. So yeah, I have to apologize because I think there's still some question that unanswered. But yeah, I think we already passed the I don't know, is it still possible for you, Rosie, to answer, or you have
I might need to go quite soon.
Yeah. Oh,
but could I answer some questions afterwards, like on email or something? I don't know if.
So, Mbak Isti, the question, Mbak Isti, the question in the chat or the question section, can you later on send the transcript to me? Also, I will send you the Rosie email, then you can send it directly to her, also along with the document that you want to send to her. Yeah.
Yeah, and I can do references as well if people want.
Okay. Then also maybe for those who have not been answered the question, please write down or in the question chat box your email. So then Mbak Isti, when she got the reply from Rosie, then she can forward it to you. Yeah. And we are very sorry because the time is limited, so we cannot answer everything. It's very fascinating, and as you see, Rosie, the curiosity of our researcher on and also the research is getting traction, and yeah, it's really
yeah. It's great. Thank you for having me.
Yeah, thank you very much, Rosie, for sharing with us. It's really with pleasure, and also for me, I also have question, but I don't want to here any longer. So with that, thank you, and I want to return the session to the moderator, Mbak Isti. Please, Mbak Isti.
Thank you very much to Dr. Rosie Drinkwater for the interesting sharing in this webinar. Hope you not hesitate to share to us again about your experience. And thank you very much to Dr. Radian Pratama as moderator. And now I would like to greet audience again by Bahasa Indonesia to continue our webinar. Untuk para peserta, selanjutnya akan diinformasikan mengenai pertanyaan yang terbaik untuk mendapatkan door prize. Nanti akan dikomunikasikan oleh panitia. Kemudian izinkan saya untuk menampilkan terkait layanan yang ada di Saraswanti Genomic Institute.
Mbak, bentar sebelum karena because Rosie will have to leave soon. Ada yang mau tampilkan nggak, Mbak, kayak sertifikat gitu untuk beliau? Atau mau dikirim aja?
Ada sertifikat via personal gitu.
Gitu ya. So Rosie, there will be a certificate also from us, but Mbak Isti will send it directly to you. So if you are in a hurry, then you can leave because after this there is a like introduction for the services from the SGI.
Okay. Yeah. I will probably have to run.
For the sharing with us, and and we hope to learn from your expertise again in the near future.
Yeah, thank you very much for having me. Have a good day, everyone.
Good day, Rosie.
Very
Thank you.
much. Have a good day.
Bye.
Pleased to meet you.
Okay, so Mbak Si, please continue, Mbak.
Terima kasih Pak Radi. Terima kasih. Berikutnya, teman-teman jangan live dulu ya, karena untuk para peserta nantinya akan mengisi daftar hadir, karena itu yang akan bisa digunakan untuk mendapatkan e-sertifikat. Izinkan saya untuk menampilkan layanan terkait pengujian sequencing yang ada di Saraswanti Genomics Institute. SGI DNA XQ Service itu merupakan layanan mengenai DNA extraction and quantification service. Di sini peneliti bisa menguji sampelnya yang membutuhkan layanan untuk ekstraksi. Kemudian ada SGI DNA barcoding yang menggunakan untuk menggunakan primer universal berupa CO1 atau COX1, sitokrom C oksidase 1. Ini digunakan untuk pada region animals sama beberapa tista. Kemudian pada penggunaan di fungi menggunakan Primata sRNA, dan kemudian untuk tanaman rubisco, dan untuk prokariot, identifikasi prokariot itu 16 sRNA. Biasanya digunakan untuk identifikasi pada spesies, sehingga umumnya sampel berupa raw sampel, kemudian diekstraksi hingga mendapatkan hasil filogenetik menggunakan layanan DNA barcoding. Lalu untuk layanan berbasis platform NGS, ada Symbios, Saraswanti Microbiome Solution. Di sini menggunakan metode shotgun metagenomics, basisnya NGS platform. Kemudian ada SGI Bacterial WGS Service, Bacterial Whole Genome Sequencing, dan platformnya pun NGS. Lalu kami juga bisa melakukan Human WGS Service, dan terakhir ada NIPT. Ini buat deteksi non-invasive prenatal testing untuk ibu hamil. Untuk lebih jelasnya, para peneliti ataupun teman-teman bisa menghubungi langsung ke nomor admin Saraswanti Genomic Institute untuk bertanya-tanya terkait mengenai sampel, kemudian bagaimana layanan yang tepat, karena SGI sendiri atau Saraswanti Genomic Institute menyediakan paket layanan dengan harga yang kompetitif dan berkualitas. Jadi teman-teman atau para peneliti jangan sungkan untuk menanyakan informasi terkait harga, kemudian term pengujiannya, lalu penggunaan alat yang kami gunakan. Kemudian izinkan saya share screen terkait link kehadiran. Tapi sebelum itu, saya ingin menutup acara ini dan mengucapkan terima kasih pada para peserta yang sudah hadir di webinar Genomics Talks yang ke-12. Semoga tetap termotivasi untuk terus belajar, mengembangkan riset penelitian, kemudian terus bersemangat untuk mengembangkan ilmu pengetahuan yang ada di Indonesia, dan tetap tertarik mengetahui lebih lanjut terkait Genomics, kemudian ikut serta di kegiatan akademik, salah satunya adalah mengikuti webinar Genomic Talks yang ada di Saraswanti Genomic Institute. Kemudian untuk para peserta tertarik bisa bertanya informasi lebih lanjut kepada admin kami. Dan jangan lupa para peserta untuk mengisi daftar hadir ya. Terima kasih kepada Bapak Rahadian Pratama sebagai ekspertas bioinformatik di Saraswati Genomic Institute. Terima kasih atas bantuan dan kerjasamanya. Semoga Bapak tidak pernah bosan ya Pak. Ya, untuk di webinar selanjutnya. Kemudian ini share screen terkait ini email yang bisa dihubungi para peserta, pertanyaan ataupun terkait inquiry mengenai layanan yang ada di Saraswati Genomic Institute, serta ini kontak yang terhubung melalui WhatsApp admin kami. Nah, ini pengiriman untuk sampel yang nantinya apabila ada inquiry sampel yang mau dikirim bisa dikirim ke alamat kantor kami.
Mungkin saya izin menambahkan sedikit Mbak Isti ya. Jadi teman-teman Bapak Ibu sekalian yang mau melakukan sekuensi tapi mungkin belum yakin atau butuh apa ya semacam bimbingan, bukan bimbingan ya, mungkin konsultasi ya, itu bisa kontak juga ya ke Mbak Isti ya.
Iya, benar, bisa.
Kita untuk bioinformatika bisa custom. Jadi itu sebenarnya dari yang tadi standarnya mungkin metabarcoding, oh saya punya sampel seperti ini, gimana bisa analisis dan sebagainya, itu tinggal disampaikan aja. Jadi tidak ada yang benar-benar baku dan yang lain ini nggak bisa. Tapi kita karena di Indonesia supportnya juga mudah, mau main data masuk ke SGI di admin juga mangga ya Mbak Isti ya.
Ya, betul, bisa sekali.
Bisa diskusi langsung gitu, jadi nggak perlu jauh-jauh ya. Saya kira itu jadi tidak perlu khawatir, kontak aja langsung. Pokoknya kontak dulu nanti gratis kan, nggak bayar Mbak Isi ya, gratis itu ya, konsultasi dulu gitu ya.
Konsultasi sebagai bagian dari layanan kami untuk mensupport customer sehingga bisa memudahkan untuk diskusi yang interaktif dari layanan yang ada di kami beserta yang cocok ya, layanan yang cocok untuk support penelitian dari teman-teman semua.
Gitu. Jadi kontak aja, aman. Bioinformatikanya bisa diurus.
Ya. Kalau untuk yang tadi pertanyaan para peserta juga langsung ke email ya Pak ya? Atau tadi sudah saya catat semua ya Pak di PowerPoint, soalnya hanya saja karena ada pertanyaan yang dipilih, jadi saya tidak tampilkan semua, tapi di PowerPoint ini saya sudah merekap semua ada berapa belas.
Saya kirim nanti emailnya Rosie, jadi Mbak Isi bisa kirim langsung email Rosie atau lewat saya juga boleh, misalkan ini aja.
Terima kasih teman-teman semua, terima kasih Pak Radian beserta rekan dan Dr. Rosie juga. Semoga kedepannya bisa menampilkan Genomic Talks yang lebih luar biasa lagi. Hari ini adalah hari yang luar biasa, perdana bagi Saraswati Genomic Institute untuk menampilkan webinar yang mengundang narasumber dari luar negeri, Jerman. Tentunya teman-teman pun memiliki insight yang luar biasa, mungkin bahkan ada yang termotivasi untuk kuliah lebih lanjut, dan kita juga bisa kerjasama untuk layanan pengujian lain. Nah, untuk lebih jelasnya bisa berkomunikasi ke kontak admin kami. Saya tutup, terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Terima kasih Mbak Isti. Ya, jangan lupa absen.